The effects of atrazine on frogs.
Questions. How does atrazine affect frogs? How do frogs change genders? What happens when they change gender? Abstract. In the natural world, frogs have the ability to change genders in order to balance the male-to-female ratio in a population. This is called sequential hermaphroditism. By doing this, frogs can repopulate despite having an unequal population of males and females. Sequential hermaphroditism can be influenced by chemicals like atrazine, causing sequential hermaphroditism by increasing the activity of aromatase, which is an enzyme that turns androgens into estrogens, male into female hormones, and vice versa. The introduction of these chemicals on a larger scale often happens when fertilizers diffuse into bodies of water. Hypothesis. Adding atrazine to the frog tank will cause the African dwarf frogs to change genders, in other words, to undergo sequential hermaphroditism.
In this experiment, we compared the effects of atrazine on female African dwarf frogs. Our control group consisted of an average fish tank, water, 2 female African dwarf frogs per tank, a heater, thermometer, filter, and decorations. The experiment group had all of the same variables except the implementation of atrazine, the independent variable. We checked in on the frogs every week and documented to see if there was any phylogenical change, the dependent variable. The two genders are distinguishable because male African dwarf frogs have white glands underneath their arms, and females don't. A female African dwarf frog is also bigger. Additional information. We purchased 4 female adolescent African dwarf frogs, Hymenopterus boeckeri. This is because African dwarf frogs are able to perform sequential hermaphroditism and are relatively cheap. We ensured their gender after purchase, and let them acclimate to the water for an hour. We also made a mixture of 1 tablespoon of atrazine to 3 tablespoons of water. With this mixture, on a biweekly basis, we added 0.5 tablespoons, equivalent to 7.39 milliliters of mixture to the tank, and tested pH in the water, after to make sure the water levels were not harmful to the frogs.
Safety measures. In order to keep our water safe, we measure the pH levels, nitrate levels, carbonate hardness, and general hardness levels. We also used a thermometer to make sure the temperature was at the right amount.
We gathered the following information for week 1. In the control tank, the frogs had female-sized white glands and were larger in size for youth. Similarly, in the test tank, they had female-sized white glands and were larger in size for youth. For week 2, the control tank had, once again, female-sized white glands and were larger in size for young adult frogs. In the test tank, they were also female-sized with white glands and medium in size. For week 3, the control tank remained similar, with female-sized white glands and larger in size for adult frogs. In the test tank, however, they were still female-sized white glands. However, 1 frog died. They were smaller in size. From our experiment, we have learned that atrazine has little effect on frogs and may possibly influence sequential hermaphroditism if there was more time. Due to atrazine having little to no effect, we do not know if the frogs change gender or in what way besides the size and male reproductive gland. We do not know what happens when a frog changes gender except for maybe the increase in territorial aggression. All of the weeks remained largely the same except for week 3 in the test tank. It was a catastrophe for the experimental group as one of the frogs ate the other's limbs. This we attribute to territorial aggression possibly caused by the change of gender, though we cannot draw it out conclusively. With this, we can conclude that our experiment effectively tested our hypothesis, but more time was needed to see if the frogs fully changed. After studying 2 separate tanks of African dwarf frogs for 4 weeks, one without any atrazine and one with, we have come to the conclusion that sequential hermaphroditism is a long drawn-out process. We have examined these frogs for 4 weeks and have yet to see significant enough change to prompt the assumption of sequential hermaphroditism. However, we do believe that atrazine could have played a part in the territorial tendencies within the test tank. Other studies also highlighted the change in territorial patterns which was witnessed. A study from 2002 believes that gender could possibly be a cause, though in our case, the results are inconclusive. For further research, we will expand our test group to get more detailed data. We would also like to note the difficulty to maintain a balanced tank because of multiple factors. Because of the frogs' aquatic nature and the struggle to gather data in the future, we would hopefully have more equipment to better gather data and create more illustrative graphs.
Sources.